Okay, I did my student project on human rhinovirus, which I from now on will refer to as HRV or just rhinovirus because it's kind of a mouthful when you're talking. But this is the virus that causes what everyone knows as just the common cold. So, human rhinovirus, the virion is made up of an isohedral capsid made up of VP1, VP2, and VP3 proteins. It is an unenveloped positive single strand RNA virus with a VP4 protein anchoring the genome inside of the capsid. There aren't any spike proteins, but it binds to either the ICAM1 or LDLR receptor of the host cell with the canyons, which are made up from VP1, VP2, and VP3. So human rhinovirus infects usually the nasal epithelia of its human host. And now I will just go through all of the steps. So for attachment and penetration, human rhinovirus attaches to the respiratory epithelial tissue using the canyons created in its isohedral capsid by the structural proteins VP1, VP2, and VP3. These canyons attach to the intercellular adhesion molecule 1, which, like I said before, ICAM1, in 80% of human rhinovirus, which categorizes them as the major group. And then the other 20% of human rhinovirus uses the canyons that bind to low density lipoprotein receptors, which is what I said was LDLR, which categorizes them as the minor group because it's only 20%. And then once HRV attaches to its host cell, it is engulfed by the cell via endocytosis, and the virus is then within an endosome and fully within the host cell. Moving on to uncoding, the low endosomal pH triggers the encoding of HRV genome, and its positive sense single-strand RNA, which is still accompanied by the VPG or VP4, those two are kind of interchangeable, is released from the isohedral capsid made up of, again, VP1, VP2, and VP3. A slightly acidic pH allows for the dissociation between the binding receptor and the virus, and then the pH of 5.6 or less allows for the release of the positive sense single-stranded RNA genome. The viral genome causes the bursting of its capsid as well as the endosome, thus releasing the RNA genome into its host cell's cytoplasm. And then for biosynthesis and assembly, since HRV is an unenveloped or like naked positive sense single strand RNA genome, it is immediately ready for translation once it's inside the host cell's cytoplasm. So there are two parts of biosynthesis: viral genome synthesis and viral protein synthesis. In order for the RNA genome to be copied into more RNA genome for its new virions, the enzyme RNA-dependent RNA polymerase or replicase is needed. So replicase is provided by the virus to carry out viral genome synthesis, and then viral protein synthesis is conducted by the ribosomes within the host cell interacting with the positive sense single-strand RNA, which produces the VP1, VP2, and VP3 to form the viral capsid, as well as some other non-structural proteins, which aid in genome replication and assembly. After biosynthesis, the individual products of biosynthesis are reassembled into a new HRV virion, and then these new virions exit the cell via lysis or bursting and go to infect other respiratory epithelial cell tissues. All right, and next we will go on to some interactions that it has. Sorry, I lost my place. Okay, so I guess we can just start with number one. Okay, sorry. Airway epithelial cells and human rhinovirus. So airway epithelial cells are the first to interact with human rhinovirus and are the virus's doorway to infect the host, binding the plasma membrane glycoproteins to gain entry, which is the ICAM1 and LDLR. In response to the infection, airway epithelial cells launch an innate pro-inflammatory immune response. And in a pro-inflammatory immune response, mast cells encounter the pathogen and release a cytokine, usually histamine, which triggers an effect on localized blood vessels. The histamine causes vasodilation, and thus blood begins to leak from the blood vessel containing various leukocytes, which attack the pathogen. And this initial inflammation produces the symptoms that immediately come to mind when you think of the common cold. Moving on to number 2, human rhinovirus and neutralization. So in a study done where subjects were voluntarily infected with HRV, some presented with the antibodies needed to fight off that specific serotype before they were even infected, which means that at some point in their lives before this study, they were infected by that specific serotype previously. So in this experiment, antibodies, which were likely IgM or IgD, targeted the virus and attacked it via neutralization. So neutralization, as we all know, is when the antibodies surround the pathogen, neutralizing its ability to attach to human cells and gain entry. Moving on to number 3, human rhinoviruses and natural killer cells. So upon infection of human rhinovirus, one of the innate immune responses of the host is to activate natural killer cells. And natural killer cells are activated by interferons released by the infected cell. Consequently, natural killer cells come to the site of infection and release a cytotoxic mediator and cytokines, which puncture the infected cell, causing it to die. And then later, macrophages appear to the scene and clean up any remaining parts of the dead cell. In the interest of time, I think I'm just going to move on and leave out 4 and 5. Okay, so this is the study that I did part 4 of my project on. So it was done on just over 5,600 students aged 6 to 17 with allergic asthma. And they did this study by reporting weekly for 90 days to see if treatment with omalizumab lessened the infection rate, illness, or exacerbations of their allergic asthma. So just for like the sake of everyone understanding what I'm talking about, an exacerbation is the same as an asthma attack. So mucus samples and reports on whether or not the individual had an asthma attack were taken each week, like I said, for 90 days. Okay, so this key figure shows that omalizumab reduced prevalence and viral shedding in mucus. And both of these figures have a p-value of less than 0.5. So we can trust that they're statistically significant. So as we see here in both graphs, the purple bar represents the placebo group and the pink bar represents the group that was treated with omalizumab. So here in the graph A, I guess, yeah, bar graph A, we see that omalizumab caused less viral prevalence, which just means, like, in my understanding of the study, it's like whether or not the individuals had symptoms. So the symptoms you think of when you think of the common cold. And then on the right, we show that it also decreased the number of individuals who had peak viral shedding, which is when, even if you don't have any symptoms, it's whether or not the virus is detected in the mucus samples that you gave. All right, and then finally, the questions I would have for the researchers who did this study would be, would the treatment of omalizumab be beneficial for the general public, even those without allergic asthma, just as a medication to lessen the duration of viral shedding and human rhinovirus prevalence? So like, if just anyone was treated with this, would we either like not have as severe symptoms of the cold, maybe not have symptoms at all, and maybe it would just get rid of it? I'm not really sure what that would be. And I guess that would be my question. And then additionally, could this medication prevent asthma attacks during peak prevalence seasons, such as like allergy season or any other time that would cause respiratory inflammation due to something that was not allergic asthma. Finally, I have slides of all of my sources. And if anyone has any questions, feel free to email me. And that's it.